Everyone, and a very warm welcome to the Florey. Delighted to see so many people here in person, and also very pleased that we're joined online by many people who are watching from home. My name's Professor Peter van Wijngaarden, and I'm very proud to be the Executive Director of the Florey. I'd like to start this morning by acknowledging the traditional owners of the lands on which we're meeting today, the Wurundjeri people of the Kulin Nation, and I pay my respects to the elders past and present. So the Florey Talks, of which this is the first for 2026, are a series of talks where you can learn the latest research insights into brain and mental health conditions. A key element of the talks is to draw on the wisdom of those with lived experience of these conditions, and we're really excited about the amazing lineup of speakers that we have for you today. So it's our hope that these talks can precipitate conversations this morning, but also conversations in our wider communities. And so with this in mind, I strongly encourage you to participate in the discussion today, but also to provide us with feedback, because we want these talks to be informative, welcoming and engaging. So it's now my great honour to introduce your host for this morning, Professor Tony Hannan. Tony is the head of our epigenetics and neuroplasticity group at the Florey. His research team explores how genetic and environmental factors combine to influence predisposition to specific brain disorders. So we look forward to a very engaging and thought-provoking conversation this morning. And on behalf of all staff and students at the Florey, I'm really delighted to welcome you all here. So please put your hands together for Tony.
Great, thank you, Peter. As Peter said, my name's Tony Hannan and I lead the Epigenetics and Neuroplasticity group at the Florey. My research team explores how genes and environment combine to influence predisposition to specific brain disorders, including some of those that we will focus on this morning. We use these discoveries to find new ways to prevent and treat these brain disorders. So welcome to the first Florey Talk of the year, as Peter said, and as Peter mentioned, they were previously known as the Florey Public Lecture Series, but I think the Florey Talk better captures exactly what we're here to do. And we welcome also the many of you who are joining us virtually. This is being live streamed. And I'd also like to thank all of you who are wonderful supporters of the Florey as you help to make our scientific discoveries and medical research breakthroughs possible. Today we're talking rare diseases, and specifically rare brain diseases. According to the Lancet journal, approximately 300 million people live with rare diseases. There are more than 7,000 types of rare diseases. Individuals living with rare diseases are often a neglected and marginalised group, especially those in low-income and middle-income countries. Around 80% of rare diseases have a genetic cause, almost 70% of which are present in childhood. About 95% lack approved treatments. The average time for an accurate diagnosis is 4.8 years. Devastatingly, about 30% of children with a rare disease die before the age of 5. So I'll pause to let you consider that shocking statistic. And we'll hear later from Mel and Deanna and Tanya with lived experience about how these diseases impact families. At the Florey, our teams work across a number of rare brain diseases. We acknowledge that this talk highlights only a few examples, but it forms part of a broader commitment to recognising and supporting the importance of ongoing focus, collaboration and advocacy across all rare diseases. Today you have the chance to hear more from key experts who are involved with rare disease research and discovery, or are connected to a broader rare disease community, including Mel and Deanna. These are all individuals endeavouring to better understand, prevent and ultimately produce greater patient prospects and outcomes for those living with rare diseases. We are privileged to have these two members of our lived experience community joining us. As I mentioned, Mel Anderson from the PURA Foundation and Deanna Carpino from the Australian NPC Disease Foundation. As I mentioned, Mel and Deanna, as well as Tanya West, have kindly contributed to the wonderful video presentation that you will see later. But first up, we're pleased to have 4 excellent Florey speakers giving you key updates on their current body of work and what this means for the rare brain diseases domain. Dr. Carolina, or Carol Gubert, is a research fellow who leads the microbiota gut-brain axis team in the Florey's epigenetics and neuroplasticity group. Dr. Gubert's research focuses on gene-environment interactions and the microbiota gut-brain axis in brain disorders, including Huntington's disease and schizophrenia. Next will be Dr. Sonali Reisinger, an early career researcher in the epigenetics and neuroplasticity group at the Florey. While her current work includes advancing our understanding of the transgenerational effects of viral infections on brain and behaviour, today she will present our work on neurofibromatosis 1, or NF1. Dr. Sarah Gordon is head of the Florey's presynaptic physiology group. Her work focuses on determining how dysfunction of presynaptic proteins, which play a key role in transmitting chemical messages in the brain, contributes to the development of neurodegenerative and neurodevelopmental disorders. And finally, the 4th scientist speaking will be Professor Reid, who is lead of the Epilepsy and Neurodevelopment Research Priority at the Florey. He is a translational neuroscientist and leads a multidisciplinary team that investigates the molecular and cellular causes of epilepsy. As I hand to our first speaker, Dr. Carol Gubert, please be assured that there will be ample time for questions at the end of the presentation section. We will have a roaming mic available at that time, but please note we are unable to provide medical advice today. Thank you very much, Dr. Carol Gubert.
Morning, everyone. Thank you very much, Tony, for this lovely introduction. Also, thank you for the opportunity to be speaking here today. It's really a pleasure to talk to this audience and also to people online. So, I would like to first introduce you to my grannies. In addition to them having the cutest great-grandchild, yes, that's my son, they have also something else in common. A phrase I've heard countless times growing up. Finish your beans. And boy, we eat a lot of beans in Brazil. And as usual, they were not wrong. Beans are amazing. Just 1 cup has about 15 grams of fiber. And you know who else likes beans? Microbes. There is a distinct microbiome in pretty much each niche of our human bodies. The skin, the airways, the eye, and also the gastrointestinal tract. And if this is news to any of you, I'm very glad to be the one breaking it, because in my opinion, this is one of the coolest biological things that we know. One of the biggest discoveries that in the past decade happened is that we know that these gut microbes interact with the brain. And actually, we have to think what happens when this communication goes wrong. And we know now that disturbances in the microbiome gut-brain axis, they are linked to a wide range of diseases, not only to the gut-related ones like IBS that you can see here, but also pain, psychiatric disorders, and neurodegenerative disorders. And here at the Florey, my team has added Huntington's disease to this list. We were the first to show that the gut microbiome of people living with Huntington's disease is different from controls. And also, we were the first to show that this also occurs in HD mouse models, giving us great opportunities to really understand how this relationship occurs in Huntington's disease. But let's take a step back. What is Huntington's disease? Well, Huntington's disease is an inherited brain disorder, meaning that a parent can pass it to their child in what we call autosomal dominant way, which means that if one parent carries the gene, each child has 50% of inheriting it. The disease affects parts of the brain that controls movement. As you can see here, Huntington's disease has a very characteristic trait, which is this involuntary dance-like movements known as chorea. Huntington's disease is the most common monogenic neurological disorder. The onset of HD goes from 2 to 60 years of age, having a juvenile and an adult onset journey, with the latest being the most common one. After diagnosis, people with Huntington's disease die 10 to 15 years after getting diagnosis, which means that this is a devastating disorder that rapidly progresses, and it is fatal. The symptomatology of HD is actually quite complex. In addition to these motor deficits, there are also psychiatric symptoms, cognitive impairment, and gut-related issues that can occur up to 20 years before the onset of the motor symptoms. And there is still no disease-modifying therapies available yet. So with all of this in mind, we asked a simple but important question. If the gut microbiome is involved, could we target it to delay the onset of the symptoms? And to test this, here at the Florey, we used a few different approaches. We started with a very good proof of concept approach, which is the fecal matter microbiome transplant. So we took microbiome from healthy animals, we transferred this into HD mice, and also we used environmental approaches, which are lifestyle-based approaches. We introduced exercise and environmental enrichment to mimic a healthier environment, strategies that we already know modulate positively the gut microbiome. We also changed their diet. We fed these mice high-fiber diet and prebiotics, which essentially give the microbes the nutrients that they thrive on. And all of these interventions did change the gut microbiome positively, and they also, all of them, enhance cognition in this mouse model. With the latest two, enhancing gastrointestinal function and increasing also ameliorating affective behavior and also improving motor function. And if you're wondering where we can find all of these little treats for the good gut bugs, here they are. Have a look. They are all everyday foods. And yes, the chickpea beans are up there. So here at the Florey, we show that the gut microbiome disruption is part of the Huntington's disease phenotype. But instead of only adding an extra issue to the pathology of this disease, we look this as an opportunity for intervention with actually broad applicability, because cognitive enhancement would be beneficial for a wide range of brain disorders. And something I'm very passionate about, we are studying things that are accessible, affordable, to be suggested as adjunctive treatment, making even closer to benefit the families. And our next steps are particularly to understand the mechanism by which these interventions has these amazing outcomes, but also moving forward to clinical trials. We showed all of this in mice. Now we need to move to human studies to really show that these interventions work in this population. And before I finish, I'll leave you with my take-home message. Thank you very much. Sonali.
Thank you, Carol, for that fascinating talk. Good morning, everyone. Hello to everyone at home. As Tony mentioned before, today I will be talking to you and giving you a little bit of insight about our work in a mouse model of neurofibromatosis type 1, or NF1, and particularly how we study cognition and behavior in this mouse model. I'll start by giving you some background about this disorder, NF1. So it's considered a relatively common rare disease. In terms of incidence, it's just under 1 in 3,000. And it comes about by the mutation of one copy of the neurofibromin gene. This can occur spontaneously or when it is inherited from the parents. The main symptom complexes have to do with the function of this gene as a tumor suppressor and its expression across most tissues of the body. So the key symptoms relate to unrestricted cell growth and tumor growth in many tissues of the body, including the skin, the eyes, and the bones. However, and this is what we're interested in at the Florey, there are also many brain-related symptoms of NF1. And these include anxiety and depression, sustained attentional difficulties and other cognitive difficulties, as well as many patients being diagnosed with ADHD, autism, or sometimes both. And these particular brain-related symptom complexes don't have specific treatments available. Therefore, the long-term aim of our project is to improve these particular treatment possibilities for cognition and behavior in patients with NF1. And we use a mouse model to address this question. So here at the Florey, we're able to do touchscreen-based rodent cognitive testing. And this occurs, just like you see on the left-hand side here, in small chambers that are fitted with touchscreen tablets, just like iPads. And these are actually very similar to the way neuropsychological tests are carried out nowadays in humans with tablets. So here we're interested in looking at sustained attention, since NF1 patients have difficulties with this. To go into a bit more detail, I'll move this mouse, of how this test works, we train the mice to respond, in this case by nose poke and not with their hand, to a particular stimulus called the S+, and at the same time we train them to inhibit their response to 4 other types of stimuli. And these are shown very rapidly. So as you can imagine, this is quite taxing for both mice and humans, and the mice take some time to learn this. However, we also have a trick up our sleeves. So when they give a correct response, we actually reward them with strawberry milkshake. And when they give an incorrect response, they get nothing. So this might surprise you, but the mice absolutely love strawberry milkshake and will work very hard to get this reward. And eventually they learn this task. Before I continue with our results, if you'd like to know more about the background of why we did these touchscreen tests and more about our work in NF1 and also see a few videos of the mice performing the tests, I'd point you towards a YouTube channel run by the Flicker of Hope Foundation, who partially funded this work. And this is run by Zoe Petropoulos, who actually has lived experience with NF1. So on top of interviews with researchers, she also has many really inspiring videos about her experiences. So I encourage you to check that out. You can find it via the QR code here or by searching Flicker of Hope on YouTube. So back to our mouse model. The mouse model that we use actually has a genetic situation exactly like the human, so 1 mutation in the neurofibromin gene. But what happens when they're tested in the touchscreen chambers in the rodent continuous performance test? The combination of results we see is actually quite interesting. So under normal conditions, they actually perform quite well and receive a normal amount of strawberry milkshake. However, when we add distractions, so in this case we add different types of stimuli on the right or the left of the main stimulus, which is usually shown in the center of the screen, then they actually perform far worse. And this was very interesting to us because this actually mirrors the situation that individuals with NF1 show in many of these neuropsychological tests, where when the cognitive load increases, the attentional performance decreases. So this suggests that this is actually a good mouse model to study the attentional deficit seen. So what are the next steps in this project? First, we still have a lot of data to analyze between me and my students. These touchscreen tests give us a lot of different data that we can look at. And we can use this to then formally compare the performance of individuals with NF1 and our NF1 mice to really determine how similar they are in these types of tests. We can then use this task to test new potential treatments to target the cognitive and behavioral symptoms in these in individuals with NF1, or first in the mouse model. And of course, long term, we would like to translate our findings into humans and help look at new treatments for these symptom complexes, as well as study the neurobiological mechanisms that underlie these parts of the symptomatology. So that's it from me. I will pass on to Dr. Sarah Gordon.
Thank you very much, and welcome everyone. I'm going to talk to you today about a syndrome that's very near and dear to my heart. I've been working on it for over a decade now. And I'm going to tell you about a sort of a story of discovery, how we discovered one of these rare brain diseases. I want to start with some of the faces of Baker-Gordon syndrome. These have been compiled by the Baker-Gordon Syndrome Foundation, which is a great foundation that's connecting families and researchers worldwide. So I'm going to talk to you about what is Baker-Gordon syndrome, how do we get here, and what are we doing to help now. So how did Kate Baker and I discover this syndrome? Like all sort of good things that started over a cup of coffee, Kate travelled up from Cambridge. I was working in Edinburgh in the UK at the time, and we were talking about an entirely different project. But then she told me about this child she had in the clinic, Lucas. And Lucas had a really sort of interesting story. He first presented at the clinic with floppy baby syndrome. And from there went on to develop developmental delay. So he first sat up at 4 years, took his first steps at 10, still can't talk, but definitely can communicate. And then went on to develop abnormal, uncontrolled movements, and then would also have these outbursts of sort of highly agitated behaviour, would go from these sort of calm states to sort of unprovoked agitated behaviour. Now, you don't have to be an expert in, brain physiology to sort of understand that if you look at a brain activity signal, that a child with Baker-Gordon syndrome has highly abnormal brain activity. And this set of symptoms was really not unlike any other disorder that Kate had seen. And this is because Lucas didn't have any seizures, no overt seizures. So really abnormal brain activity, but no epilepsy. So she did sequencing on Lucas and his parents and found the first ever variant in the SIT1 gene. And so that's where I got brought into the story. She asked me, do I know anything about SIT1? And I happened to be working on it at the time. And what I can say is SIT1 is probably one of the most important genes in the human brain. So it's absolutely essential for your brain to work properly. What it does is it triggers the release of the chemical neurotransmitters that allows your brain cells to communicate with each other. So we wanted to find out whether variants in SIT1 would sort of change the way brain cells work. And we used our brain in the dish model at the Florey, where we have brain cells that we get from mice, and we can plug and play different genetic variants that are found in humans into these brain cells. And then we're able to look at these neurons, brain cells, talking to each other in real time. And what we found is that variants in SIT1 cause an impairment in the release of these chemical neurotransmitters, meaning that the brain cells aren't talking to each other properly. So once we had sort of discovered what was causing the disorder, we're now able to use this model system in a range of different ways. So to start off with, we're trying to improve the diagnosis of Baker-Gordon syndrome. So we've only known about this syndrome for about 10 years now. So we're still getting contacted by clinicians all over the world who are asking if this particular variant in SIT1 might cause the disease. And so we're able to put those variants into our model and see whether they do in fact impair the ability of the brain cells to communicate. But that's not just true for Baker-Gordon syndrome. SIT1 doesn't act in isolation. It works with a whole community of other genes that are all working together to make sure that the brain cells are communicating properly. And so now we're looking at all these other related genes and seeing whether variants in those ones also impair neurotransmitter release. We're now using this model system to try and understand better how this dysfunctional communication actually leads to the specific set of symptoms in individuals with Baker-Gordon syndrome and other related disorders. And importantly, we're now able to test therapeutics. And this is really important because right now there's basically no therapies, no effective therapies for Baker-Gordon syndrome or many of the other related disorders. So we can try targeted therapies in our model system. From here, we're really excited to have some new assets in our toolkit. So what we're doing is getting skin cells from individuals with Baker-Gordon syndrome and related disorders, turning them into stem cells and then turning those into neurons in a dish. So those are human neurons that were able to grow in a dish, and they contain the exact same genetic variant that was present in the individual that we got the skin cells from. And then we can test precision therapies. So we can work out whether we're able to reverse the defects that are occurring in those brain cells, and essentially leading us towards what we hope will be a disease-modifying therapy in the future. Thank you. Pass across now to Professor Reid.
All right, so thank you so much for coming to discuss what is a very, very important disease collective. So what I'm going to talk to you about today is mouse modelling of the rare genetic brain disorders. Specifically, I'm going to talk about, I'll use the, so people can see that. Oops. Can people still see that? Yeah, that's good. That's right, I'll just chat along. So what are the DEs? They're the most severe forms of epilepsy. They start early in life. Children do not respond to current drugs. There's significant developmental delays, and there's a high risk of sudden unexpected death in epilepsy. They're a collective group, and it's very easy to sit there and say, this is what the disease is. But what it truly is much better said from a patient's perspective, and you will hear from Mel in a minute. This is little Fern, you can see here, and this is her dad's description of the impact that the disease has had on her. So the impact it's having on this child is omnipresent, spectre, single family. It's life-changing in the truest sense. Relentless seizures, constant lengthy hospital, sleep deprivation, stress, inability to work. So the impact on these patients and the carers is massive. This is a quote from Mel, who you will hear from later, and her daughter has PURA syndrome. It took more than 20 drugs to find a drug that actually partially helped seizure in Sarah. Sarah still has seizures to this day. I think Mel texted the other day that she had 21 seizures. So even though we've got this whole scree of drugs, we don't know which ones to use in Sarah's case and for PURA in general. And not only that, a lot of these medications actually have significant side effects, which can often be worse than the seizures themselves. We have to do better. We do. This is the problem. The developmental epileptic encephalopathies relate to developmental delay in epilepsy, but there's over 900 genes that can cause this disease. So there's 900 ways that you can break the brain for you to end up with epilepsy and developmental delays. What that means is there's probably about 900 ways that you need to fix the brain in order for that disease to be dealt with. So although it's a collective, it's really quite a complex collective set of diseases that need to be thought about as an individual genetic cause. So how can we go about starting to unravel that complexity? And the way that we do it in our laboratory is to develop these DE models. Now, the important point here is that these DE models are made by CRISPR Technologies, which is a technology that's been around now for about a decade. And what it means is that we can actually take the variant that's in Sarah or Fern and put it into the mouse. So these mice have the same genetic problem as the children. And the big advantage of that is that we can generate a large number of mice and do a whole lot of things on those mice where there is only one Sarah, one Fern. So it gives us that capacity to start to understand the disease and also maybe to test a whole lot of different drugs, and I'll show you that in a second. So we have generated a lot of different mice. You can see the different genes on the top. The importance of that is not so important. What is important about this slide is that each one of these different mice have a slightly different epilepsy, all right? And that's important because that means we're starting to understand that complexity, and we've got model systems that give us the opportunity to delve much deeper. So we do a lot of that, but the other things that we do is these mice can be used as almost to screen drugs. Like Sarah went through that process, one at a time, one or two together, etc. We can, as I said, make as many of these mice as we need, and we can test all the drugs that are already on the market and perhaps find out which ones best suited. And we know that the thing works, this process works, and the reason that we know that is based on little Fern. So Fern's dad, Phil, gave me a call, and Lauren, I think, is in the audience as well, and we had a bit of a chat to her. He knew that we were working on HEN1, so Fern has a variant in HEN1, and she was having about 10 seizures, quite significant seizures per month. At the end of that conversation, it was said that she was on... something's wrong here. Sorry, there's a slide that's not come up. Can people see that now? Yes. Yeah. So Fern was on a drug called lamotrigine. Now, taking that concept that I talked about having all these different mice models and the capacity to actually record from these and understand what different medications do, what you can see here, these are anti-seizure medications you can go to the pharmacy now and buy. What was remarkable is a subset of these worked in the mouse. And the subset actually made the seizures worse. So when Fern's dad gave us a call, the last thing we had is, well, what anti-seizure medications are you on? And she turned out to be on a drug called lamotrigine, which is that middle one. And we know from the mouse that it was making things worse. So when Fern was taken off that drug, she went from having 10 seizures a month to actually being seizure-free now. So we know that the paradigm works. We know that we have the capacity to use these mice to screen. And what our hope is, in the context of PURA syndrome, is that we can use this mice to stop this, what I call medication roulette, where Sarah had to go through 20 drugs to identify drugs that may work more easily. To that end, we have made a PURA mouse. It's the one just on the thing. This is obviously not the real mouse, but it is fascinating. But we do have real mice upstairs, and these real mice do have seizures. They have the drug biomarker that we've talked about, and our hope now is that we can use this mouse to understand what drugs best suited for PURA syndrome and also potentially understand mechanisms better. So this is the vision. We don't want to just help Fern, we don't want to just help Sarah or the PURA syndromes. We want to have a collective impact across the whole 900 genes. And we've got an idea here where we have people like Ingrid Scheffer, who sees a lot of these patients. She has a patient that comes in early in their journey. We make a CRISPR mouse, as I talked about. We start to test the mouse, like I've just shown you with PURA, understand what drugs work, understand what mechanisms are, create therapies, and then take it back to the patient. So that's the vision, and the vision is to understand that complexity, that whole umbrella. I'm sorry about the technical difficulties, but hopefully the message was clear. I am going to hand over now. Are you going to do that? Yes. Great, thank you.
Thank you, Carol, Sonali, Sarah and Chris. I think that gives you a flavour of the world-class neuroscientists we have at the Florey, which stand shoulder to shoulder with the very best scientists in the world. So at the Florey, we know that the voices of people living with or closely associated with someone who has a brain or mental health condition are central to the research we do. Connecting with those who have lived experience helps guide our priorities, deepens our understanding, and ensures that the research we pursue truly makes a difference to people's lives. With that in mind, I would like to share a video in a minute and introduce you to some very important Florey supporters and lived experience voices who have so generously given their time to share their stories with us today. And then we'll be able to speak to them live afterwards. So the first will be Mel Anderson, Director and Chair of the PURA Foundation Australia that Chris just referred to, which has established which was established to raise awareness, provide education and support research for individuals diagnosed with PURA syndrome and their families. Deanna Carpino, President of the Australian NPC Disease Foundation, which aims to give hope, support and connection to those affected by Niemann-Pick disease type C, NPC, and acid sphingomyelinase deficiency, ASMD, in Australia. And a supporter and friend who wasn't able to join us today, but will be in the video, is Tanya West, whose father Graham sadly died from multiple system atrophy, a rare form of atypical Parkinson's, which progresses rapidly and for which there are no effective therapies available. So Nick from Florey IT is going to help us get this video started now, and you'll be able to see this brief video, and then I'll invite Mel and Deanna to the stage for a conversation.
My name is Mel Anderson. I am a mum to my Pura Perfect Sarah. Sarah is cheeky. She is determined. She's incredibly resilient. Pura generally presents with intellectual and physical disability of varying degrees, developmental delay, and severe epilepsy.
My name is Amy Bowen. My husband Jim and I have 2 children. We've got Christian, who is 4 years old, and a 12-month-old boy, Matthew. Christian loves to play outside. He loves to swing. Pura first became part of our family story when Christian was 14 months old.
So the Pura Foundation formed in 2023. We all came together to walk the Melbourne Marathon Festival, and we raised $15,000. That's meant that our families had the capacity to say, we've raised this money and we want this research done.
Being part of the Pura Foundation has just been the best thing for myself and my husband, especially my family. As a parent with having a new diagnosis, it can be very daunting, scary, and you might feel isolated. But there is hope. Together we can give our children a life that they deserve.
My name's Deanna Carpino. Me and my husband have 2 children. Shortly after Noah turned 7 months, we found out that his liver and spleen could be felt. 6 months on of diagnostic testing, we found out that he had Niemann-Pick type C1 disease.
Niemann-Pick disease type C is a neurodegenerative disorder that affects primarily children. Children lose the ability to walk, to talk, to think, to play, and also to remember.
I'm currently the president of the Australian Niemann-Pick Disease Foundation. Our primary goals are to support research and families in the space. We understand that it's such a long journey from drug discovery to a cure in our hands. But if it gives an additional 5 years, 10 years to someone's life, especially a young person's life, I think it's heavy impact.
So multiple systems atrophy, or MSA as it's more commonly known, is a progressive neurodegenerative disorder. It's very specific, very quick, currently incurable.
Good morning, my name's Tanya West. I live in Canberra with my family. My dad was and still is an incredible man. I have many beautiful memories of a great childhood and a great relationship with him. We first started noticing a decline of Dad's health probably 10 years before that formal diagnosis of Parkinson's plus syndrome and obviously ultimately MSA. There wasn't a lot for us to understand outside of straight Parkinson's. So when I came to the Florey and I said, this is the situation, I would love to fund some sort of medical research grant in my dad's honour. The Graham West Research Grant is about raising awareness, getting people to talk more about the more specific syndromes of Parkinson's. It's the key for early detection, prevention, treatments. It's the key for a cure, and that's what we want.
Wow, that was an incredibly powerful video, and I'd like to again thank Mel and Deanna and Tanya, who's online, for their contributions to that video. So it's now delightful to be able to invite Mel and Deanna to the stage here so we can have a conversation to follow this up. Please, Mel and Deanna, join us here. Thank you. So Mel, if I could start with you, please. Could you please introduce yourself and share a little about you and your daughter, Sarah, sharing some moments or experiences that have been most meaningful to you?
My name is Mel Anderson. I am the Director and Chair of the PURA Foundation Australia. Thank you. There we go. My name is Mel Anderson. I am the Director and Chair of the PURA Foundation Australia. I am first and foremost a mum to my PURA perfect Sarah. So Sarah was the first Australian diagnosed with PURA syndrome back in 2014. She's now 18 years old. So Sarah presented very quickly after birth with some symptoms. At 36 hours of age, she suddenly plummeted in terms of her temperature to 32 degrees. We couldn't get her warm. She had no suck reflex, no gag reflex. She was incredibly floppy. She couldn't swallow, and she struggled to breathe. The breathing monitors ran off into the night, and we knew there was something seriously wrong. We were told by doctors in the coming weeks that she would be intellectually disabled, physically disabled, clinically blind, would probably never communicate, and that we just had to accept that because they couldn't tell us at that time what was wrong. So Sarah's diagnostic odyssey took 6.5 years. And at that point, we went from 25,000 genes of interest to 20, and then finally got our PURA diagnosis. So at that time, nothing was known. It was just a gene. There was no talk of treatments, no community, no support. We were on our own. And so one of the things I had to grapple with as a mother was, how do I help her? What do I do? And I actually went to our neurologist and said to him, how do I deal with this? What do I do? He was the one that had given me the diagnostic result. And he said, Mel, you need to find the other families. You need to come together, form a foundation, start research. It's the only way you're going to get the answers that you need. So I took that as my literal roadmap, because I had no other, and I reached out on social media. I know nothing, I'm not a scientist, but my daughter's been diagnosed with something to do with PURA. Is there anyone else out there? Several weeks later, I heard from an American mum who said, my child's been diagnosed with something to do with that too. Long story short, we formed a global foundation with 5 of us in the very early days, myself and a group of parents over in the US, and that became the PURA Syndrome Foundation, a global entity that would support families across the globe. And the thought always was, once we find more families here in Australia, we'll start a foundation here. So as you heard, in 2023, we started our foundation here, and we went out to our families and said to our families, what are your concerns? What are your worries? What can we do for you? And we heard 2 areas that kept coming back. One was communication and one was epilepsy. Our children are non-speaking. We don't say non-verbal, because anyone that's heard my daughter will know she's verbal. She's just non-speaking. So our family said, we need communication research. And I mentioned this to a number of clinicians, and they looked at me and said, but they can't talk. What's the point? Well, there is a point. We need to know why. So we went to Professor Angela Morgan and we started a research project. And at that time, 2 weeks after we actually launched our foundation, we were involved in the Melbourne Marathon Festival. You saw the photos, and we've had the privilege of Florey staff also being involved in that community event. We raised $15,000 2 weeks after we launched, and that gave our families agency. What research do you want? What do you need? Because there's lots of research that was happening in rare disease, but it wasn't PURA specific, and it wasn't going to help our families in the way they needed to be helped. That was part of our job. So we started our communication research, and we're told maybe 10 or 20 will be involved. Rare disease is rare, they're spread across the globe. But what they underestimated is the power of patient advocates like ourselves. We have 104 currently in that study from 24 countries across the globe. And that's going to get us some answers that we need. The second area was epilepsy. As you heard from Chris, my daughter's failed 20 anticonvulsants by the age of 7. We ran out of meds. We didn't know what to do. And often the seizures were either increased or just simply changed because of the drugs involved. That wasn't helpful. And I remember saying to my daughter's neurologist, what do we do here? What's the best seizure? We seem to have one. What one do you want? He was horrified, but that was my lived reality. We couldn't stop these. And over the years, it's really for us become a massive concern in our community. So where do we go to do our clinical research? We had that sorted. But where do we go to do our scientific research? And there was only one place in my mind, and that was here at the Florey and with Chris Reid. Chris is known, he is a global expert in epilepsy models, in particular mouse models, and we knew from previous research attempts that's what we needed as a global community. So our research here at the Florey isn't just for families in Australia and New Zealand, it's for families across the globe. This is our global centre for PURA epilepsy research with our mouse model. And for our families, it's giving us incredible hope that maybe we can stop this seizure cycle and stop just managing the damage being done and actually create some real change.
Thank you, Mel. That was wonderful and very powerful. So now I have a question for Deanna. So living with or alongside a rare disease can be incredibly challenging. Can you provide us with some insight into the personal impact, not just for your child and your family, but the children and families within your foundation?
Was Mandy started the foundation in 2009 after her 2 children were diagnosed with Niemann-Pick type C1. And it followed a very similar trajectory of need, inspiring action, inspiring connection in the scientific community. And that need was based quite simple.
We might just swap with that other microphone. Sorry, dear. Let's try the other mic.
Hello?
Yeah.
Cool. It was a similar set of symptoms that we found with cognitive delay, which impacts more than just the individual. It changes your entire family dynamic inside your community, how you go to work, how you see different issues, negotiating between up to 40 healthcare clinicians. I mean, that's a lot of administrative coordination that needs to happen, and with lack of coordinated care sometimes, especially for those of our families in Australia living in regional areas, it's quite a big burden, quite a big change to negotiate yourself through.
Thank you, Deanna. That was lovely. Okay, now I'll ask a series of questions and ask Mel to answer first and then Deanna to the same question. So the next question was, what is one thing that would make a significant difference, in your opinion, to patients and families living with the rare disease that you focus on?
Well, that one's easy. Effective targeted treatments. That's what we need. And we need them based in neuroscience. As Chris touched on, we're living on a roulette wheel. It's trial and error approach, medication after medication. And what scares me as the head of a foundation is I'm seeing young children being diagnosed with PURA syndrome, and they're trialling and failing the same medications that my daughter tried and failed 15 years ago. But we have no clinical evidence, no scientific proof that there's any better way. So our clinicians out there are doing the best they can with a really difficult set of circumstances. And what we need is informed neuroscience to be able to say to them, even at a basic level, don't go near these meds, trial these ones first, on our way to actually finding a precision medicine that will work for PURA epilepsy. That's the bit that we're missing. As Deanna touched on, for us, trying to manage the hospital admissions, the ward admissions, the amount of failed medications my child has had, looking at the MRIs, the CT scans. She's had 26 EEGs and we're booked in for our 27th because we're spiralling out of control with her seizures at the moment. If you look at the costs of all of this over her life to care for her, governments would be better off actually investing in rare disease research. It would be cheaper long term rather than doing this for child after child, adult after adult across their lifetimes. So for me, it's the effective targeted treatments that we need.
Great. Thank you, Mel. And you mentioned you're going to Canberra soon. I hope you get in the ear of the people in Canberra that need to hear that message, because that's a very powerful and correct message. So Deanna, do you have anything to add with that question?
I think for me it also just continually comes back to the coordinated care improvements that could be done. The simple loss of time in receiving an approval letter from your primary clinician through to your coordinator for NDIS, that delay means huge things. I mean, that's a delay in funds to get that daily improvement. That could just, we throw around the word game changer a lot. Game changer for a family just means significant improvement in your day-to-day living. Now, obviously we know research takes time and absolute investment needs to happen there, but we also need to focus on what our family needs today. And I think that comes back to the issues you've had with communication, etc. So I think coordinating care is something that's actionable and can be done in the short term.
Great, thank you very much. So the next question for both of you was, how has partnering with medical research institutes like the Florey impacted you and your organisation?
For our families, it gives us hope. And some days when your child's seizing uncontrollably, hope is what gets you through. We need change, and we need to change the narrative not just with regard to epilepsy, but with regard to the ways that our governments deal with rare disease. To touch on what Deanna was mentioning with the NDIS, we've had families knocked back for communication devices because, and I quote, they can't communicate, so why do they need them? That's why. When you've got families fighting for the basics and they're not getting those basics, we need great change and we need to do something a bit different. For us, we're hopeful that our work here at the Florey, with Chris and his team, with our mouse model, is going to lead to better understanding of the mechanisms involved with PURA, better understanding of what is actually happening with this genetic condition, because we still don't know the fundamentals yet. And we're watching our clinicians and our specialists essentially throw mud at the wall and hope something sticks. And we need to do better than that in this day and age. The research and work that we're doing for PURA won't just benefit PURA, it'll benefit all rare diseases. We all support one another. When something happens in one rare disease that creates change, it has a flow-on effect. But we shouldn't be relying on our families who are dealing with the daily burden of the rare disease to be the ones having to fundraise for the research, to be the ones having to change the narrative with our government departments. We have enough to do managing our admin, as was pointed out. It's a lot. So hopefully, through working here with Chris and his team, we can start to create that change. We can start to give our families better clinical outcomes. And then hopefully over time, we can actually get some precision medicines that will do greater good.
I think that's a great message, that science brings hope and ultimately precision medicine. Deanna, would you like to add to that?
I mean, she's very good.
Hard act to follow, really.
100%. The only thing I would probably add to that, which really just runs on your theme, is around the collaboration that could potentially be happening between certain research pieces. I think that's why, like, one of our key focuses has always been conferences, not just for family connection, which is obvious, but really giving those researchers the opportunity to have a coffee, to find a new gene, to find a new therapy, that finding those connections between the different research pieces that will benefit more than one individual disorder, which we've already seen benefits in Niemann-Pick C, but I'm sure there's plenty more to be found. I mean, for me on the outside, research just seems to be a series of innovations, and I wait for the next one to hopefully apply to my own situation.
Fantastic. Thank you very much, Deanna. So the next question was, from your perspective, what challenges do communities face in raising awareness and funding for rare brain diseases, particularly given their low prevalence and limited public visibility? What approaches have you found helpful? Yeah, you go first this time.
Sure. For me, I have a bit of an issue with the word awareness. It feels inherently passive. And I feel like when people say I'm doing something for the purpose of awareness for Niemann-Pick, they don't understand the real world actions and results that happen from that awareness. I mean, it should be, I don't know what the correct term will be for it, but it is an actionable awareness initiative. When we say that we're raising awareness for Niemann-Pick C in the clinical realm, we need clinicians to not look for the common, but to look for the zebra. And if they don't know about it, it has a lot of real world implications. And same goes for fundraising, for research. there is always going to be a bit of an uphill battle. There's so many great causes, so many great disorders that need attention and need funds to make that research happen. But I have a child, I have to defend him, and I have to try, and I think that will always be a bit of a battle, especially in the ultra-rare space when there's only 30 to 40 people alive in Australia right now with Niemann-Pick C.
Wonderful. Thank you very much, Deanna. What would you like to add to that, Mel?
For us, it's the invisibility of being rare. When there's only that number and there's only 40 of us in Australia and New Zealand that we know of, and I say that we know of, because I can guarantee that they're out there. We just haven't found them yet. Rare disease by nature and by definition is rare. So we're spread across our country. We're spread across the globe. So for us, the hardest part is finding one another. We were very lucky. I was particularly lucky to be involved in my community from the day dot. So I've been involved in a community that has tried so hard to stay connected, to grab hold of one another and to make sure that as we grew globally, we were connected, we were supporting one another, and we were there. Our mid-year conference, I ran the first 3 of them for our global community. That is essential for us to connect, to bring together our researchers, to bring together our families. And for me, the awareness concept is education. Now, I'm a teacher, so that's just who I am, but it's about educating people. That's how I raise awareness, is if I can get one more person to talk about PURA, they may tell one person, and they may tell one person. And that's how you get it out there. I know a lot of people look at social media and think, is it doing a lot? But for rare disease, it's our lifeline. It's how we find one another. It's how we connect. It's how we support. And in some ways, that's one of the greatest things that we can do as patient advocates, is try and connect community. And I've always been a big believer that for us, and we call it Team PURA, and the Florey knows Team PURA now, we have our clinicians, our scientists, our families, our foundations, all coming together as one. Everyone has lived experience. And I know often when people talk of lived experience, they talk about us. But our scientists have lived experience. Our clinicians have lived experience. And that's incredibly important. Those puzzle pieces coming together are what will get us the answers that we need.
Thank you, Mel. That's a very powerful message. So I'm going to ask one more question, but before I do that, I want everyone in the audience to think about, after that, I'll be inviting the 4 scientists out the front as well. So if you had questions for any of the 4 scientists, or Mel or Deanna, there'll be an opportunity after they answer this final question, which is, what advice would you offer to individuals or families at the beginning of their rare disease journey?
It's such a unique journey, and I know that everyone tackles those initial feelings quite differently. For me, I needed a pathway. I needed to hear that there was something to do. That's how I tackle issues in general. Some people need to sit with it, and I think that's okay too. My advice would be to don't lose connection. No matter which way you sit with the feelings or with the diagnosis, just keep the pathways open. I think so many of us disconnect from our partners in that situation as well, or take ourselves away from it in order to deal with it. But at the same time, I would just encourage you to keep all those connections alive. For me, I was genuinely excited to find the foundation when I eventually did. Unfortunately, at the time, they weren't so great on social media, and it took a little bit of deep diving, and I found them in the backyard in regional Benalla, but I found them. And that was just like a hallelujah moment, because up until that point, I had been told similarly to, this is it, there's no treatments, just go home, make memories. I'm not okay with that. So that's probably my advice.
That's a beautiful message, Deanna. Thank you. Mel?
For families going through their diagnostic odyssey, or those that have just found their family and their community, ask the uncomfortable questions. We're often told this is just the way it is with rare disease, or that's just how it is with epilepsy. You're just going to have to deal with it. When my daughter first started having seizures at 14 months, we were told she'd seize every day for the rest of her life, and you just had to deal with it. No, I don't. That's not an option for me. So ask the uncomfortable questions. Find your community. This journey and this road is too long and too hard and too heavy to do on your own. Find your people. Find clinicians who will listen and hear you. Because that's the difference, hearing us. We've had clinicians that have stayed with us for 18 years. My daughter just turned 18 at the end of last year. And that's made such a difference, having people that were there and who listened and heard you. Start your own research. Don't wait for other people to do it. That's the only way you're going to get those answers that you need. And that's the way that you can create the change that you need for your community. That's what led us here to the Florey, that need for neuroscience research. This is a neurodevelopmental condition. We need specialists in neuroscience. And that's what's going to get us those targeted, effective treatments that we need.
Thank you, Mel, for that really moving message. And I'd like to ask everyone to really thank these two extraordinary women and mothers whose children were struck down by brain disease, and their response has been to set up foundations and fight back. And I think, join me. Thank you. Great, so now we get to invite our four wonderful neuroscientist speakers from the Florey to the stage. Carol Gubert, Sonali Reisinger, Sarah Gordon and Chris Reid, who are going to join us now. And now's the opportunity for anyone in the audience. We have a roving mic here, so please, in the audience, raise your hand if you have a question for any of the four scientists, but also our two lived experience individuals, Mel and Deanna. We have a question up the front, I think, to lead off with. Thank you.
Just testing. Yeah, great. Thank you. That was fabulous, guys. Thank you so much. Two questions, if I may. Firstly, AI, precision meds, what are the thoughts there? That's just a small project. And number two, in terms of, I'm an emergency physician, but in another life, neuroscience at Uni of Melbourne with Professor Darian Smith, which may be a familiar name perhaps, long time ago, with the astronomical increase in ADHD diagnosis, has there been an increase in NF1 diagnostically? I'm just curious about that. Thanks.
I can start with AI, and I think that will be a game changer. Absolutely. We're still working out how to harness the powers of it, but it's something that, particularly here at the Florey, that we've got front and centre of our mind. How can we be at the forefront of big data and AI, bring those together in order to try and accelerate our therapeutic strategies? And that comes not just from screening drugs, but also through the more sort of precision medicine sort of strategies. So, yeah.
We've had this conversation quite frequently around what precision medicine means. I think even getting what's in the pharmacy currently and working well in the children is a great start, and it's a lower hanging fruit. So that's the first step of it. Obviously, because we know the genetic cause of a lot of these diseases now, and genetic therapies are becoming quite common, we really are on the cusp of starting, and the Florey's been central to that as well. So precision medicines from a genetic perspective, from a standard of care perspective, and a repurposing perspective are real. The other point to make, which is really important from our perspective, is PURA at the Austin, who sort of specialises in these rare epilepsies, has just got a big MRFF grant to do N-of-1 trials. So we can take what we're learning here and into the clinic in this concept of N-of-1 trials, which I think, each one of those collectively will then give us an idea. So, yeah, we're getting there.
And Sonali, did you want to add anything on how our NF1 research may impact on autism and ADHD more generally?
Maybe first about the question of the incidence that you spoke about. So ADHD increasing, I think it's a difficult question because it has a lot to do with the diagnostics, right? And I think in NF1 it's similar. There are many patients who don't show that many symptoms, and they might be diagnosed just by genetic testing, which is becoming more common. So I do think there's an increase in diagnosis. But in terms of whether that is a real increase or, just us getting better at identifying it, I think that's still a difficult question.
And I wonder, Carol, whether you want to add anything on how AI and this kind of bioinformatics and these technologies relate to your research here at the Florey.
Yeah, absolutely. As I mentioned before, our discoveries of the gut microbiome brain axis is pretty new. It's 10 years. So all these new technologies are helping us to really identify the bacteria that live in our body and how it communicates with the brain. So definitely all these new technologies are helping us to really dive in mechanism, as I mentioned before. And this is what we are really trying to focus on here at Florey.
Great answers. Thank you very much. Hands up, anyone in the audience who had a question for anyone on the stage, for any scientist. We have a lady there in the middle. Thank you.
Thanks very much. This has been fascinating from many angles. I was just wondering more about the brain in a dish. I'm very curious about how that works. I'm wondering if you could explain a bit more about, just how that all works and what you can see from it.
Yeah, we take brains from generally embryonic mice because they still have the most growth potential. We very, very gently separate them into single cells, and then we put them in a dish and just let them mature. And they will form the same sort of connections that are forming in our brain. And we've got some really cool little assays that we can use. A lot of them are fluorescence-based, so we use microscopy. And essentially the cells flash every single time the brain cells are talking to each other. So we're able to look at how frequently they're talking to each other. We're actually seeing them talk, flashing at each other, able to see that in real time and get lots of different measures of how effectively they're able to communicate. And then we just plug and play different genetic variants into that system and see whether we can whether it makes things worse or affects it in different ways, I guess. So, yeah.
That's really cool. Sorry, tiny follow-up question. You said you use mice. Have you done any trials using human, induced pluripotent stem cells, like eye neurons?
Yes, so that's what we're doing now. So we've gone from, the mouse brain cells are really, they're great because they mature really quickly, actually. The issues with using the stem cell derived approaches is that it's a lot slower, but they are human neurons, so that's the benefit, right? Is you're looking at actual human neurons in a dish, and so we're employing all the same strategies that we used in mouse neurons. We're now putting them in human neurons and doing the same sort of things.
Peter was going to say something.
Yes, our Florey director, Professor Peter van Weingarten.
Just wanted to add another point that the modelling of these complex disorders requires a variety of different approaches. So we've heard about mouse models, we've heard about mouse neurons in a dish, we've heard about stem cells that we take from skin and using Nobel Prize winning innovation, turn those with a variety of different chemical cocktails into any class, pretty much any class of neuron now. So we can study those in isolation, but we have a diverse range of expertise here in the Florey, including world-leading research in bringing a complex array of different cells together, because a brain is more than just one class of neurons. It's an array of different neurons with vasculature, with the supporting cells, the so-called glia or glue of the brain, into these complex communities of cells. And each of those different levels of models have different strengths and different advantages. They all generate vast amounts of data. They're all very, very labour intensive. And so we have world-leading infrastructure to support that sort of approach. So we are very proud to have a stem cell robot, which can do the job of 10 or 12 people, but it can do it continuously, day and night. So we can do large volumes of these cells. So for instance, for our motor neuron disease research, we can take skin cells from 150 different people with a variety of different forms of the disease. We can scale those up, and then we can test vast libraries of existing drugs or novel chemical compounds to see which ones work. And you imagine the vast array of work that involves. We can use robotic technology and even automated microscopes to facilitate that. So it is that diversity of approaches that really helps us push the envelope on these complex diseases.
Thank you, Peter. That was a great summary of how Florey is going from molecules to mind to prevent, treat, and eventually cure a whole range of brain disorders. So did we have any other questions from the audience? Anything you wanted to ask for anyone out the front? We had a gentleman here in the front.
I have a couple of questions, if that's possible. First to Carol. Carol, thinking about the gut-brain axis, presumably a two-way axis. So the microbiome change you're seeing, is this a Huntington's brain talking to the gut?
Yeah, that's a great question. It is a two-way communication, and we are still in the infancy of understanding how it communicates both ways, but we have a couple of ideas of how this might be happening. We have one neural communication, the vagus nerve, that literally communicates the gut and the brain both ways. So we have afferent and efferent neurons that directly communicate gut and brain, but also we have the metabolites that the microbiome release that goes through the blood, that also goes to the brain, but also we have some HPA, so hypothalamus, so cortisol can also communicate back to the gut, so it's definitely a two-way communication.
Have you asked the question, does Huntington's affect the enteric nervous system?
That's another great question. We are starting collaborations to start looking at it, because of course, ENS is the second brain, as we call, some even call the first brain. We don't have data yet on that, but people are already starting to study this relationship.
If you imagine a broken protein like PURA, wherever it's expressed, it's going to have an impact. Yeah. So the work that we've done has shown impact in the retina, for example, for the HN1 channel, which is highly expressed. So for a lot of these diseases, it's not just the epilepsy that's the problem, it's a whole scree of different things, including the gut, and that's becoming clearer and clearer. So at least from a precision medicine perspective, if we're fixing something in the eye and the brain, we should also be fixing it in the gut. So that's where precision medicines are really going to start to come into their own if we can deliver them across the brain.
That's very helpful, thank you. Chris, I had a question for you. Given that the condition you look at broadly may have 900 causes, is it time to re-examine the possibility, the feasibility, the acceptability of Crelox and genetic manipulation?
You mean in humans in vivo?
Exactly.
So the idea of Crelox, like CRISPR, somatic gene editing of adult cells, is that one direction?
It's certainly on the horizon. A lot of that technology is a little scary because when you make the change, you've made it. And if it goes wrong, you can't reverse it. And so there's aspects around that we need to consider. But for sure, I mean, there are a number of different genetic approaches that are now being taken. Overexpression of RNA, ASO technologies, which I think the Florey lead in the world, some of which have actually got to clinic and showing quite good promise. So I think it's going to be like most things. It's going to take a multi-pronged approach, and not every disease is going to fit each of those. So, but yeah, CRISPR editing is definitely on the agenda worldwide. Not the stuff that we're doing, but it is on the agenda.
Great. I think that's a great note to finish on it, and there will be morning tea outside, so please stay on, and you'll be able to ask any further questions to these six wonderful people afterwards over morning tea. Also, this has been live streamed and it's recorded, so please get your family and your friends to go to the Florey website. This will be, when it's finalised, it will be made available on the Florey website, on our YouTube channel, and so it'll be accessible if you've missed anything today. Of course, there's a lot of other information on the Florey website or the Florey social media channels. We provide a lot of information there as well. So it just leaves me to thank these six extraordinary people for sharing their passion and their dedication and their talent today. So please join me in thanking Mel, Deanna, Carol, Sonali, Sarah and Chris. So morning tea outside, please join us.